Hi, thank you all for tuning in to my presentation, which is part of the Eastern Washington University Research and Creative Work Symposium of 2020. I'm Jenny Walke, an assistant professor in the biology department here at Eastern, and I'm really excited to be here to share my research and my life story that goes along with that research with you all today. I'd also like to thank the organizers for giving me the opportunity to talk as part of this symposium. So this image here is agar art. Instead of using paint, a paintbrush, and a canvas, agar art uses microorganisms, inoculating loops, and agar plates to create art. And this is one of my favorite agar art images because it combines two of my research topic areas, which are bees and microbes. And I'll come back to that in a little bit. But first, I'd like to share with you guys one of the greatest lessons that I've learned over the years being involved in scientific research. And that is that creativity is just as important in science as it is in art. And so according to this sign here, which stands outside of my daughter's elementary school and was posted several months ago, as the word of the month was creativity, being defined as using imagination to solve a problem. And again, I just think that creativity is key to solving scientific problems as well. Okay, so most of life is invisible. This is the tree of life showing the three domains of life: bacteria, archaea, and eukarya. And as you can see, just the tiny tip of one of these branches is actually visible life. Things like plants and animals that we can see with the naked eye. And the rest of life is invisible, microscopic organisms that are all around us. And these microbes do really, really important things for our planet. And nearly all multi-cellular organisms, so again, think plants and animals primarily, are host to symbiotic microorganisms. And these constitute their natural microbiome. And here are just a few examples. Maybe you're familiar with Finding Nemo and the anglerfish here at the bottom that has a light organ that's filled with bioluminescent bacteria that it uses to lure prey and attract mates. So a really important association between the microbe and the animal, and really important for its survival. So in addition to survival, these microbes do a lot of other really important things for the hosts that they're associated with, ranging from influencing their development and behavior, their immunity, nutrition, metabolism, really important functions. And so again, it's a crosstalk between the microorganisms and the chemicals that they're producing that are being sensed by the host's systems, whether it's the immune system, the nervous system, and so forth. And it's often, these relationships are bidirectional too, whereas the host systems can influence the composition of the microbes that are associated with that host. And an interesting study that just came out last fall found that the microorganisms and the composition of the microorganisms in the guts of humans can influence the effectiveness of vaccines. And in this case was a vaccine for influenza. And it was a study where they altered the microbiome with antibiotics, and that altered the immune response as well. So really important interaction going on between the gut microbiome and the immune system in humans. And so really, you know, the moment that we're born, the moment that plants and animals begin to develop, we're constantly going to be colonized by a host of microorganisms. And these microbes have been around on our planet a lot longer than we have. And so we really have evolved in the presence of these organisms. And so we are animals in a bacterial world. And to kind of broaden that, we're really, you know, plants and animals in a microbial world, because bacteria are just one type of microorganism. And so my research in particular studies the microbial ecology in several systems, primarily amphibians and honeybees, but I've also been involved in studies looking at the microorganisms associated with birds and lizards, and more recently, since arriving at Eastern, looking at wetland microbial communities and their importance in carbon cycling and responses to climate change. Also thinking about the microbes that are influencing disease transmission and vector transmitted diseases, such as the West Nile virus that's transmitted by mosquitoes. And also looking at the relationship between the clam microbiome and metal pollutants in their environment as well. And so for the purpose of this talk, I'm going to focus on the amphibian and the honeybees and some of my research in those systems. And I'll start with the amphibian system. And yes, I was able to find auger art for the frogs as well as the bees too. Although I cannot take credit for creating this wonderful masterpiece here. Okay, so we'll start with the frog system. So unfortunately, frog populations and salamander populations around the world are experiencing declines, and many of them are even extinct. Biodiversity is experiencing loss across the world, but amphibians are thought to be the most vulnerable vertebrate taxa, as nearly half of all amphibian species are either extinct or threatened with extinction. And this is due to partially things like habitat loss and pollution, but one of the main reasons that amphibian populations are declining around the world is a fungus that infects their skin and causes the skin disease. And this fungus is called Batrachochytrium dendrobatidis, or BD, as I'll refer to it in the rest of the talk. So this fungus, like I said, it infects the skin of amphibians, and it disperses in water, and it has a motile zoospore stage that comes into contact with amphibian skin, and it insists down into the skin, where it then turns into the zoosporangium that produces more zoospores that can disperse and reinfect that same individual, or disperse and infect a new individual. And so this is the life cycle of the fungal pathogen. However, when these fungal pathogens, these zoospores, are coming into contact with the amphibian skin, they're not alone. There are lots and lots of other microorganisms on the skin, including lots of bacteria, that are also there. And so what I'm interested in with my research is the interaction between the amphibian host as well as this fungal pathogen and the other microorganisms on the skin and how they all interact. And so this research, I actually got involved as an undergraduate student at James Madison University in Harrisonburg, Virginia. And here I was interested in understanding the diversity of microorganisms on the skin of amphibians, as well as their role in potentially influencing disease susceptibility. And so a lot of the work that I did there was culturing bacteria and then testing each of those bacteria against the chytrid fungus in these laboratory assays. So here you can see the cloudy area on the plate is a lawn of BD, and the bacteria of interest that I streak and look for these clearings around the bacteria. And so in this case, the Pseudomonas species is producing chemicals that are able to kill and inhibit the growth of BD in these laboratory assays. And so the next thing to do was to try to identify some of these chemicals that are anti-BD. And so we have worked to be able to do that as well. And then after that, what we're interested in is seeing is whether or not these interactions and the ability of these bacteria to kill the chytrid fungus in these lab petri dish assays, can we replicate that, and is that also observed in live animal hosts? And so with this experiment, which was done on the mountain yellow-legged frog of the Sierra Nevada mountain, that's one of these species that is experiencing declines. And so what we did with this frog species in the lab was, some of them, we infected them with BD. And so you can see their survival over time in the orange line. So over the 20-week experiment, nearly all of them died when they were exposed to the chytrid fungus, BD fungus, in the lab, which is what we expected to see, that because they are a susceptible species. However, if we inoculate these frogs with this anti-BD bacterium and then expose them to BD, they all survived, which you can see in the green line, that they all survived throughout the course of the experiment. So this was really exciting, that this, temperate amphibian species and this temperate microbial species was actually able to protect this amphibian in the lab. Unfortunately, when my colleagues and I did an experiment on a tropical frog species, the Panamanian golden frog, which is one of the species that's thought to be extinct in the wild. When we did this similar experiment, unfortunately, the probiotic bacteria, this anti-BD bacteria, did not protect this species. And we thought that that was because it's a temperate microorganism and a tropical amphibian species. And so this bacterium just didn't persist long enough on the skin in order to be protective. So what did we do? We went to the tropics, and Panama in particular, and looked for tropical anti-BD bacteria that could potentially be used to protect tropical amphibian species. And this work was done with collaborators across a couple different institutions, and we were based out of the Smithsonian Tropical Research Institute in Panama, where we sampled amphibians and cultured lots and lots of different bacteria that could potentially be probiotics for tropical frog species. And so related to this, we tested all these microbes against BD in the lab, and this is one of the results that we found. So some amphibian assemblages, or kind of groups of different types of amphibians, are able to coexist with the fungus in nature, whereas other amphibian assemblages are experiencing declines. And so then when we quantified the amount of protective bacteria in the two different groups, we found that those amphibians that could coexist in the presence of the fungus, they had a greater proportion of those individuals had protective bacteria compared to the declining assemblages. And so this is very similar to herd immunity theory, where if a certain, proportion of a population is protected, it kind of protects the whole population. So this was really exciting to see in our work in Panama. And then the same pattern was also observed in California with the Rana muscosa mountain yellow-legged frog that we had done that laboratory experiment on as well. So those coexisting populations had higher proportion of individuals that were protected compared to those that are declining in the presence of this fungus. And so at this point in my career as a scientist, I had done my undergraduate, and I was working for a couple years as a research technician after my undergraduate. And so at this point, my mind was blown in terms of the power of microorganisms and the relationships that they can have with their hosts and influencing disease susceptibility. And so I knew that I wanted to go on to get my PhD and continue doing research at this point. And I just have to say too that I my relationship with my undergraduate mentor continues today, even 15, 20 years after I started working with him in the lab at JMU. And so just keep that in mind too for the undergraduate students who are listening today, is that you never know how long some of the relationships with your professors might end up lasting. Okay, so I went to Virginia Tech to get my PhD, and a lot of my work was actually based out of Mountain Lake Biological Station. So even though that is owned and run by University of Virginia, which is the in-state rival of Virginia Tech, they were very kind and let me conduct a lot of my dissertation research at Mountain Lake. Also during this time of my PhD, about halfway through, I had my first daughter. My husband was in one of those images at JMU helping me collect salamanders in the field, and here we are now with our first daughter, Claire. And I just have to point out too that these two pictures are of my daughter, kind of at around, I think she was like 3 or 4 months old in the tie-dye, in front of this enclosure that's in the pond, if you look at that, which was part of an experiment that I did with salamanders, where we were tracking the skin microbiome of these salamanders over time. And those same enclosures are in the background of the picture next to it, where she was probably about 2 or 3 years old there. So this, again, just to point out how long science can take sometimes as well. So she's a great field assistant there, and is also still a great field assistant now. And so one of the main things that I was interested in with my PhD was to further understand variation in the skin microbiome of amphibians and how that relates to variation in disease susceptibility, because different species vary in their susceptibility to BD. And so I was interested in if that was related to variation in their skin microbiome. And so I surveyed a lot of local amphibian species in Virginia. And this image here this graph here is showing the similarities among microbial communities. So basically what you need to know here is that each point represents either an amphibian species from a newt or bullfrog in this case, or a water or pond substrate sample. And the closer points are together, the more similar their microbial communities are compared to points that are further apart. And so you can see that, the bullfrog and the newt points are separated and grouped together by species. And that is different from the environmental microbes as well. And so even these two different amphibian species that are coexisting in the same environment and exposed to the same environmental microbes, they have distinct microbial communities. And that might explain why some species are more susceptible to disease than others. And also, just take this moment to relax and think about the tropical environment, because what I want to talk about next is what's known in ecology as this latitudinal diversity gradient. And so again, thinking tropics and closer to the equator. And what we find is that for most macroorganisms, again, like things that you can actually see, there's this pattern where closer to the equator, there's high biodiversity. And then as you get closer to the poles, the diversity decreases. And so this is a general pattern that's been shown in a lot of macroorganisms, including amphibians. So if you look at the diversity of amphibians, it's typically greater along the equator, and then it decreases towards the poles. And so with amphibians, it kind of follows that gradient that is typically found in macroorganisms. But interestingly, what we found is that if you look at the microorganisms, that the microbial communities on the amphibians, is that the pattern was actually opposite to this latitudinal diversity gradient, such that the skin microbiome of tropical frogs had low biodiversity, and the cooler climates had higher diversity. And you can kind of see that pattern in this figure as well. If you look at the x-axis and kind of the cooler temperatures, and the richness, which is a measure of diversity, is greater in the cooler temperatures than in the warmer temperatures. And so it's this opposite pattern with the microorganisms compared to what we have typically seen in ecology with the macroorganisms. And all of this research on amphibian skin microbiota is thinking about using these bacteria as probiotics for wildlife conservation. And interestingly, the author Ed Young, in his book, I Contain Multitudes, discussed some of our research on amphibian skin bacteria and probiotics as a way to kind of set the stage for his topics on human probiotics as well. And so that was kind of interesting that he was able to pick up on some of our research. And again, just kind of making this connection with humans. So the purple bacteria here, it might look familiar just a few slides back. And this is one of those microbes that it produces these anti-BD bacteria that can, kill, or produces anti-BD chemicals that can kill this fungus. And so now there are clinical trials going on with this amphibian skin microbe in human clinical trials to treat human fungal diseases. So again, you'll never really know, sometimes you'll never really know how your research might end up and benefiting humans as well. Okay, so I graduated with my PhD. And again, just a shout out to my support, my graduate advisor, and my lab mates, and my family, and my husband and daughter, Claire, and parents. They're just being so supportive, really important in allowing me to be able to kind of start my family and continue with my research as well. So thank you to those out there who continue to support me as well. And after I graduated with my PhD, I did some teaching at Roanoke College, which is a small college in Virginia. And it was really exciting to be able to further kind of incorporate students into my research and continue doing that. But that was a short-term position. And then I decided I wanted to do some more research. And so then I wanted to kind of broaden my my study system and kind of broaden from the amphibian skin microbiome and kind of use my knowledge that I gained in that system and some of the methodologies and apply it to a new system. And so that is what led me to work on the honeybees as well. And yes, those are my daughters in the bottom panel there that I was able to get their pictures published in a research journal article. And so to kind of broaden my research, I did a postdoc at Virginia Tech, went back to Virginia Tech to do postdoc research. And that is where my husband and I had our second daughter, Nora, there. And so again, just really thankful to the support to be able to continue research and do exciting things like paint bees, as you can see here, me doing experiments with honeybees to look at the role of the gut microbiome of honeybees in their health, because they are super important and are experiencing declines and population declines as well. And so kind of the broad theme of this research is to think about how environmental factors like pesticides and nutrition and disease, how that impacts the gut microbiome and the structure of it, so who's there and how much of each member of the microbiome is there, and then the function, what are they doing? And also vice versa, how the microbiome can influence, the ability of the bee to detoxify pesticides that they might encounter, or acquire nutrients from the limited food sources that they have available, or to be able to resist diseases. And so this is kind of the broad framework for my research now, which is funded by the National Science Foundation. And with this research, there's the potential to be able to use honeybees and their gut microbiome as a model system to study the more complex and more difficult to track and experimentally manipulate human microbiome too. And so one of the experiments that I did when I was a postdoc at Virginia Tech was expose different bees in the hives that the bees are in, to various chemical pesticides that the bees are likely to encounter. And so I mixed these pesticides in these pollen patties and administered, and of course I had controls as well. And then I did dissections of the bee guts in order to quantify their gut microbiome. And one of the main things that I found was that certain types of bacteria, including Lactobacillus, which are thought to be really important for the bees, and you can see here the amount of Lactobacillus in the purple parts of the bars, the Lactobacillus decreased in these 3 pesticides that I tested in comparison to the control. However, if I added this probiotic bacteria mixture that's actually commercially available to beekeepers to the bees, then that restored the level of Lactobacillus similar to controls. And so again, there's an interaction between the environment and the microbiome, but that could potentially be mitigated by probiotic bacteria to maintain healthy bees. And so then after my postdoc research, I landed this amazing job here at Eastern Washington University, where I've been for several years now, and continue my research both in bees as well as amphibians as well. And so we have bees at the campus garden behind the red barn, as well as on top of the new pub green roof. So we use those for research as well as education and outreach to share with the broader community the importance of bees as pollinators for our food crops, as well as natural ecosystems as well. And so one of the projects I'm working on now is continuing looking at the pesticide concentrations in the field without experimental manipulation to see if there's a correlation with the gut microbiome. And so I've sampled 6 different sites across the Eastern Washington region, and preliminary results suggest that the bees with certain chemicals, the chemical pesticides in them, different amounts of them, those have different microbiomes than the bees that do not have these particular insecticide chemicals. And so we're really interested in kind of combining our field results and our laboratory manipulative experimental results to really try to understand the interaction so that we can potentially increase communication between beekeepers and the agricultural industry, and make sure everyone is aware of when and what is being applied to crops and so forth. And I'm also continuing the amphibian research here in collaboration with the Idaho Department of Fish and Game, where we are looking at populations across the Idaho northern panhandle to look at the relationship between BD infection and the skin microbiome, again, in a natural environment. And so we continue to do that, as well as work at the Turnbull National Wildlife Refuge outside of Cheney. Also, as part of the amphibian project, I am co-founder of this group called SNAPS, which is the Student Network for Amphibian Pathogen Surveillance. And again, this is a way to incorporate research and surveillance of diseases into the classroom and have students be able to participate in surveillance and contribute to understanding and monitoring for these pathogens across the world. And so this organization is an international effort to try to kind of coordinate institutions, primarily undergraduate institutions, to with a goal of, you know, amphibian conservation and disease surveillance. And so hopefully now you guys are, you know, realize the power of the invisible world, and realize that, you know, yes, there are definitely disease-causing microorganisms. We certainly know about that now. But the reality is that the majority of microorganisms are harmless, and in fact, many are beneficial and do really critical roles for humans, for animals, and our ecosystem overall. And lastly, just thinking about, you know, the future and microorganisms' role in the future, I really do think that microbes are going to be key to human health, and thinking about personalized medicine based on our microbiomes. Also, you know, agricultural health, and more broadly, thinking about environmental issues and how microorganisms are going to be really critical for helping with pollution and metal contaminants and plastic degradation. Plastic is a huge problem on our planet, and we're finding that some microorganisms can degrade the plastic very rapidly. And so we might be able to use microbes to help with that as well. And so thank you all for listening, and thank you to, again, my support network along the way, and my collaborators and students that have been involved in this project, and funding agencies as well, including Eastern Washington University and the National Science Foundation. I hope you enjoyed this, and thank you.